Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Professor Robert Siegel. He's at Stanford in the Microbiology and Immunology department. We're going to talk about he's a teacher, so he's not doing research so far as I know, but he's a commentator for a lot of news outlets, especially lately on coronavirus. He's been teaching for many years and has quite an extensive knowledge of various viruses that affect humans. So, Robert, thanks for coming.
It's my pleasure. Thank you for inviting me.
Yeah, how long have you been studying viruses and what got you interested in them years ago?
Well, I first started teaching about viruses about 40 years ago. And I think what got me interested was I was actually teaching a course about cancer, the biology of cancer. And we talk about some of the major causes of cancer. And we kind of classify those into 4 groups. And those are basically chemicals, radiation, viruses, and genetic predisposition. So right off the bat, viruses became very important, in terms of explaining how cancer works.
Yeah, just a quick question about cancer. What viruses have been established to cause what kind of cancers? I've heard of like HPV, but what else?
Well, there's only a small number of viruses that have been definitively linked to cancers, but they've been linked to some of the most important cancers. And there are estimates that somewhere between that makes up upwards of 15 or more % of the total cancer burden. The most important ones are HPV, which is associated with human cervical cancer, as well as cancers of the genitalia of men and women, so anal cancer, penile cancer, laryngeal cancer, because that's the larynx has similar tissue. Another really important one is hepatitis B, which is linked to liver cancer. Hepatitis C is also linked to liver cancer, and then there's several of them that cause cancers that are less common throughout the world. So there is a herpes virus, 2 herpes viruses that are associated with cancer. One is Kaposi sarcoma associated herpes virus, or KSHV. And that is associated with a number of cancers that occur in, for instance, people infected with HIV. There's also another herpes virus, Epstein-Barr virus, that's been associated with several types of cancers, including Burkitt's lymphoma, which is another type of cancers of the immune system, and nasopharyngeal carcinoma. And there's also some of the other sort of less common cancers. There's a cancer called Merkel cell Merkel cell cancer, and it's caused by a polyomavirus called Merkel cell polyomavirus.
Yeah, and I guess there's also a class of viruses that have endogenized into our own DNA that we carry with us forever, right?
Yes. So it's been estimated that as much of our genome is devoted to sort of cryptic retroviruses and other viruses as the amount of DNA that's devoted to actual protein encoding region. So these cryptic viruses, they don't actually, as far as we know, come out in humans. So they're sort of pieces or remnants of viruses that got embedded in the genome long ago.
So in your more recent years of study, I know, again, you've been teaching for a very long time, 40 years, but, as an aside, are you doing active study into various viruses? That's also how you balance your intellectual workload, or, in addition to teaching, which is, is plenty. What do you do as well in the field of virology? What do you study?
Well, the main thing I focus on is teaching and public education. So I also work for a number of non-governmental organizations that do prevention work. So, for instance, I'm involved with a number of organizations that work in Africa to prevent HIV infection. I've been involved with a project that actually was looking at the incidence of malaria in Papua New Guinea. I'm currently working with some students on a review of the asymptomatic and pre-symptomatic transmission of the current coronavirus, and finishing up a chapter on the classification of viruses. So the teaching and the education part keep me quite busy.
Very good. Well, I'm sure it's on everyone's mind, the current coronavirus, but with previous viruses or, ones that are still healing, HIV, that have had a huge impact, what are some of the big learnings that you see society has figured out about that, or about Ebola, or about measles, or about, again, viruses that may have been with us for quite a while now?
Well, we're continuing to see a huge impact from both the viruses that we know and also viruses that are emerging, and that's one of the key points. We've also, as we've gotten better and better insights into these viruses, we've actually figured out some ways to deal with them. So vaccines are not particularly new, but we've now developed new techniques for developing vaccines, and some of those have been important, for instance, in the development of a vaccine against Ebola. We also, when I first started teaching, there were essentially no effective drugs, or very few effective drugs against viral infections. And since then, there's been the development of a number of effective drugs. Some of the most sort of remarkable treatments have been produced for HIV, in which we can't eliminate the virus from people's body, but we can squash it down to a level in which it doesn't cause significant health effects and it's no longer transmissible between individuals. We also have some really remarkable drugs for hepatitis C that actually are able to eliminate the virus from most people's body. So these are examples of sort of great success stories. I think there's a lesson there, and that lesson is that in most cases, the drugs have required learning a lot about the particular viral proteins and targeting those viral proteins with specific drugs.
And coming to the current SARS-CoV-2 virus, I keep hearing we don't know. What do you know that we know now after about 5 months of experiencing it? Is there any significant knowledge that's been gained about it, and in what regard?
So I would emphasize the first point that you mentioned is that it's kind of remarkable, in some sense, what we don't know, but also what we do know. So there's some very basic things that we don't know that are knowable. And part of the reason we don't know them is because there's kind of a rush to get answers, and sometimes that actually slows us down quite considerably. An example of that is that right now we're doing a lot of testing, and in some places they are actually following up positive tests with daily calls, and some people turn out to be, they're at risk, but they turn out to be asymptomatic. But for instance, we don't know what percentage of those asymptomatic individuals will become symptomatic. Okay, that's something that's very basic. It's very necessary, but there are problems in terms of HIPAA laws, and there's problems in terms of manpower, and there's problems in terms of getting approvals to doing that kind of research. And so there's some very, very basic things about transmission and about what we might call viral dynamics that we still don't really know the answer to. In terms
I have a quick question here. Why is there a problem with manpower? It doesn't make sense. If this was a world war, it would be all hands on deck. Why has no nation or no group of nations said, we're going to put forth X $100 billion and we're going to get volunteers and we're going to study this thing and get answers now? And ethics aside, it's causing trillions and trillions of dollars of consternation and economic damage. Why would there be not enough manpower, not enough resources?
Why indeed? I think you brought me in here to talk about viruses, and I think you've now entered the realm of politics. So I think the answer is political, and it's not really virological. There are a lot of answers that are knowable, and in some cases, the answers are easily knowable. So, for instance, before the very first case arrived in the United States, there was a ship called the Diamond Princess, and there was an outbreak on that ship. And it occurred to me, now I believe all the research should be voluntary, but it occurred to me that that was a perfect opportunity to study those individuals every day, to offer them tests, to find out, who was actually going to become infected. Now, not mandatory, but just, offer them and put the effort in. At that point, we would have known what the incubation period was. We would have known, if there are asymptomatic individuals and what percentage were. That study wasn't done. And to a certain extent, that study still hasn't been done. We are now 12 million people into this epidemic. So those things are knowable, but the studies really haven't been done. And I think it's for political reasons.
Well, what else would you say is knowable, and what would be, very difficult to find out?
Well, I think that given our situation, there's a lot that there's more that we can know than that we can't know. So we talked about what you can't know and why that's significant. We didn't actually talk about some of the things that are known. So one of the things that is kind of remarkable is that is the speed at which some of the molecular biology of this virus has been revealed. So even by the time the first case was announced outside of China, so this was basically the last day of 2019. That's why it ends up being, COVID-19. Even at that point, the cause of the pandemic was known, and the virus had been sequenced. Okay, that's remarkable and unprecedented that at that speed we would have determined that information about an agent that had never been seen before. And enough was known about it that we knew that it was very similar to another agent that had been studied, namely SARS coronavirus. And so those are insights. At this point, the genetic makeup of this virus is very well known. We know every single letter that goes into its genome. The proteins that it encodes are known. They have been basically their three-dimensional shapes have been determined. We have tracked changes in that genome. So there's actually quite a bit that we do know.
So so if we know the genome, I guess we would know what the mutation rate is. I know there's been, probably 15,000 plus sequences, cataloged by GISAID and others. So, has anyone reviewed them to see, how much it mutates, if it mutates, and what's the phenotypic result of the mutations? Any studies there?
So there's some confusion about this issue, because it turns out that viruses actually mutate at a very high rate, and we actually know the mutation rate. And basically, given the mutation rate, probably every cell that generates new coronavirus has new viruses with mutations. So there's the sort of the replication mutation rate, but most of those viruses, don't come to, we don't see them. They essentially don't make it. Now, one thing that's interesting about coronavirus, just as an aside, is that most viruses that have RNA as their genetic material, such as coronavirus, aren't able to act. They have a very high mutation rate because they don't have a proofreading function when they copy their genetic information. But coronaviruses do. So that's something that's kind of unique about coronaviruses. So they're making these mistakes, but then we do see changes in the composition of the sort of virus population over time. So that's not really the mutation rate of the polymerase. It's kind of the rate at which the new variants are taking hold in the population, and that also occurs at a significant rate. It's not as high as the actual mutation rate, but we're seeing the virus evolve over time, and it evolves fast enough that we can actually track where different viruses originated. So if somebody gets infected with a virus, we can determine, did that come from the China cohort, or did it come from the European cohort? And then we can get much more fine-grained than even that.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
So, okay. So I guess it sounds like no one's really characterized what the mutated strains are doing so far, or the errors from the strains. I mean, have they, or no?
Yeah, so people are looking very carefully at the characteristics of these strains. Once again, the biggest thing that we don't know is how these mutations act in human populations. And then again, that's a thing that's knowable, but really, hasn't been studied. So again, you were talking about putting large quantities of money into this. To a certain extent, as much as possible, we should be getting the sequence from every single individual, the predominant sequence from every individual who's infected. And then we can actually combine that together to see if there are trends in terms of are some of these actually more likely to cause blood disorders, or more likely to cause very serious respiratory disease. So we could actually start making correlations between the particular sequence of the virus and the outcomes. In addition, it would be great to know actually a bit about the genetic makeup of the host, because it may turn out that certain features of the virus only show up in particular genetic backgrounds.
So what else do you believe needs to be done to help, get a handle on the viral parameters, and so that society can function and know what to do from here? Okay,
so what our biggest goal right now is to basically control the virus. So if we think about, how we would control the virus, you can think about all the things that have been talked about in controlling the virus. One is the development of one or more highly effective vaccines. Another is the development of one or more highly effective drugs, therapeutics. Another would be the hope that the virus will become milder during a different season, or that the virus will evolve toward a milder state. Another is, people have talked about, if we pray hard enough, like maybe the virus will go away. And the last one is that behavioral interventions are another way of making the virus go away. Of all of those, the only one for which we have data that is effective in controlling the virus is the behavioral interventions. And so right now, it's possible that we may have a vaccine in 6 months, or a year, or 2 years, or 5 years, but we can't be sure. It's possible we may have some very effective therapeutics, within, by the end of the year, or in 2 years or 5 years, but we don't know for sure. The only thing we know for sure is that behavioral interventions are capable of stopping this virus. So we need to sort of double down on those behavioral interventions. There's lots of things that get in the way of those behavioral interventions. They include the fact that, they have huge economic impacts, they have huge psychological impacts, they have huge political impacts. But the fact is that we need to have a long-term perspective and say, if we don't double down and get rid of the virus now, what is the long-term consequence of having to deal with this virus over the course of a year, 2 years, 5 years? What's the economic impact of having to deal with this virus? What's the psychological impact if we don't get rid of the virus?
What does getting rid of the virus, what does controlling the virus mean in terms of numbers and in terms of specifics?
Well, ideally it means we become New Zealand and we drive the infection rate to zero, and they now have resumed life as they know it, aside from travel. So that's really the goal. So the way that the first SARS virus was controlled was through behavioral interventions. There was never a vaccine. There was never a drug. And basically it was controlled and it was eliminated from the human population. So that's the ideal. Short of that, we need to become, places like Italy and drive down the rate until it's low enough that we can now institute things such as contact tracing, where we're looking for the residual cases. We have so many cases in the United States that we can't even do contact tracing because the virus is everywhere.
So what do you think is a likely 6 months or 1 year into the future, at least in the United States, let's say?
I think on the track that we're on, things will be worse in 6 months. So I think that, for instance, if we look at the development of vaccines, a typical vaccine takes 10 to 15 years to develop. Drugs take a long period of time. And even after they're developed, we have to look at the manufacturing pipeline. In the US, 6 months later, we don't have the pipeline for adequate N95 masks. We don't have the pipeline for adequate swabs, okay? So now we're talking about getting the pipeline to get, the entire population of the world effective vaccine. So not only do you have to develop the vaccine, you also have to manufacture it and distribute it. So I'm looking at a fairly long timeline. Same thing with the drugs. Now, in terms of seasonality, it's worth looking at that too, because people have, spent a lot of time sort of saying, oh, the virus is going to burn itself out. Well, we've now seen it during every season of the year because it's been more than 6 months in the northern hemisphere. That covers half of the season. And it's been more than 6 months in the southern hemisphere. And that covers the other half of the seasons. And there's no indication that seasonality is affecting this virus, at least so far. And furthermore, if it behaves like other respiratory viruses, it will get worse in fall and winter. And so that actually is not a good sign. If it behaves like flu or like some of the other respiratory viruses, that may, bode poorly for our outbreak. So what
do you think will happen when flu season returns? Do you think that is there a possibility in having studied virology that people that get flu, if they still have residual coronavirus in them, do you think that'll be a pathogenic synergistic combination of both that'll make them both far worse for you, or do you think there'll be any interaction there?
Oh, I think that's an outstanding question, and the answer to almost every question, I believe this is following the inventor of, I'll take a little diversion here. The inventor of vaccines is a guy named Edward Jenner, and he came up with a vaccine for smallpox. And his mentor was a guy named John Hunter. And in the late 1700s, he told Edward Jenner, if you want to know the answer, you have to do the experiment. And so the answer is that we need to either do the experiment or look at the data very carefully before we make those predictions. So yes, it's reasonable to think that things could get worse during co-infections with both influenza and coronavirus, but we don't really know the answer very clearly. Again, this is revealing. In fact, we could know some of this because we've already seen coronavirus during a flu season, but we haven't really gotten the data.
Yeah, and when you said that things you believe may be worse in 6 months, worse in what way? In terms of the death rate, or just number of cases, or all factors? Like, what factors specifically do you think will worsen?
Well, if you look at there's some indication that, for instance, the death rate is going down. I think that's somewhat controversial, but there's some data that's been coming out in the last week or two. The death rate's going down, but if you look at the number of deaths, it's going up, and it's actually it's going up dramatically. And so in some places, it's as high as it's ever been. The hospital population, in California, of people with coronavirus and people in the ICU, it continues to rise. And so if that happens, we may be looking at some of those things that were being considered as worst case scenarios, like in New York. Some of those are coming to play in different parts of the country now. That could get worse.
Okay.
I think I'm less sanguine, I'm less interested in making bold predictions than in saying that we need to prepare for some of these worst case scenarios. And basically the alarms that have been rung by public health officials since the very beginning of the pandemic have not been heeded. And so it a lot of this stuff is not even new. It's just people aren't doing it. Like, for instance, the need for a national plan of the sort you were talking about. So one of the editorials I wrote was a 10-point plan for creating a Manhattan Project to stop this virus. That's kind of what you were referring to. It hasn't happened.
Yeah, exactly. Yep. I don't know. I mean, do you? Well, I guess I'm not sure what to ask you. I guess I know that you can't be held to any prediction. No one can. But do you see any turning of the tide here, or do you again? What do you see as what would be a good prevention strategy from here? With our exact conditions, if you were to suddenly run the show in terms of virus response, what would you do from here?
Well, what I would do from here is I would start out with, I'd say, gambler's fallacy. Gambler's fallacy is we've already put the money on the table. We've lost this much. We can't stop gambling now. Okay. So I would say we need to start from scratch and say at this point in time, what do we need to do to control this virus? And the answer is we need to do something that looks a lot more like Italy. So the question is, if we double down for a month or so, and in the harshest way, and we can control this and sort of get to another place, we can, then I think in the long run, the total amount of suffering and death will be a lot less. So yes, that would be really painful, but I think those are the things that need to be done. You have to understand that if everybody buys in, we can stop this in 1 or 2 rounds of infection. So we can stop this in a matter of weeks if everybody buys in, okay? If everybody doesn't buy in, then those efforts may just turn out to be, sort of not that helpful, which is sort of what we did so far. They were somewhat helpful in bringing that. What they did was they stopped the rate of increase, but they never produced the proper decrease in the total number of cases. So there's from an epidemiological standpoint, people talk about the transmissibility of the virus. So on average, if each infected person infects less than one other person, then the incidence of the virus will start to decline. And the faster the lower that number is, so if each person infects 0 people, then the pandemic will stop in 1 round, okay? If they infect, a small number of people, then it'll take several rounds, okay? So it really depends on how effective our interventions are. But what we've essentially done is brought that average transmission rate to around 1. So it's sort of hanging out where it was before in a lot of places in the U.S. And some places it's still way above the infection.
Yeah, but at the same time, more and more people have been exposed to it. So we are also heading towards somewhat of a herd immunity, which will dampen the infectivity rate, if there is such a thing long term.
Okay, I think that, again, herd immunity is one of those sort of wishful thinking concepts, and I'll explain why. So herd immunity suggests that, if you have enough people who have immunity to this virus, that they will protect the remaining people, and the probability of transmission will be less than 1, and so they probably won't get infected. Okay, so what's wrong with that? Well, first of all, for measles, herd immunity is at about 90% or more. So that means that you're going to have 90% of Americans, and so if you calculate the death rate from that, even if the death rate is 1% or something like that, you're talking about millions of Americans dead. Okay, so you don't want that. Okay, now what if it's less? People have estimated that maybe coronavirus might be 50 or 70% of the population. You're still talking about millions of people dead. And then furthermore, even if we got there, it wouldn't produce herd immunity. Here's why. Herd immunity assumes that everybody is equally distributed throughout the population. But if you have 95% immune and the last 5% live together, for instance, let's say like in a nursing home or maybe in a prison, then it just takes one case in there and basically you will put the remaining people at risk. So if the people at high risk are protected, they're not really protected because unless there's 0 transmission in the general population, you haven't produced any herd immunity. So the assumptions associated with herd immunity don't actually equal the reality of who's getting infected.
I see. So, very good, Robert. We're just about out of time. What's the best way for people to and your work and to follow up from here?
Well, I have a fairly extensive web presence. You can also get a hold of me at my email, which is chickenpox@gmail.com.
Yeah, Robert, thanks for coming. I appreciate it.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.